Hey guys, I hope everybody's had a nice long break and you're ready to get back into this 211 information and finish the semester strong. We don't have that much longer to finish up the semester, so hopefully it won't be that big of a challenge that we're switching to online. But I'm going to provide some extra resources to make sure that we are capable of finishing the semester strong. So I hope that you have had a nice rest, and I know I have, but at the same time, we've been working really to get this stuff moving. So I'm happy that we can start with our first lecture. So this is going to be a recording that you can look at over and over again if you want to. You can pause it, rewind it, and see the information that you want to look at again, and just pick and choose and listen to it multiple times. So this is going to stay available till the end of the semester. So use it however you'd like to. I'm also going to add some, let me get the files over here. I'm also going to add some new information to Blackboard. So first off, I'm going to start adding some, I'm going to call these outlines. They're a little bit more detailed notes than they are outlines, but this is going to help give you some extra information so that you can study at home. Also, make sure that you're hearing these recordings properly, because I expect for you to take notes while you're listening to these recordings, just like you would in class, and then study your notes just like you would normally for our test. But now I'm going to give you some of these extra notes, and for this chapter, I've got about 7 pages or so of these notes, and so I'm going to put these up under a new folder in course content before you know it. So be on the lookout for those new outlines that you can use while we're going through this information, and you can kind of highlight, or you can just kind of add to the information as well if you want to print them up and do that. I'm also going to add our usual study questions. Here's the study questions, for example, for this chapter. Again, I apologize. I don't know why there's this grid in the background of these files, but there is. And so, but again, the study questions, you're going to use these with the notes that you take in class or while you're watching this recording, and the extra notes, extra outlines that I'm going to share with you in Blackboard here coming up. And while you're studying for the test, I want you just to study like we normally have, and then the test is just going to be on Blackboard instead of being in class. The tests are going to be timed, and so you're not going to have enough time to sit there with your notes open necessarily and answer all the questions. So you definitely have to prepare, because you're only going to get about 45 seconds per question. And that's what the rest of the biology department is doing as well. So we're all kind of agreeing that this is how we're going to test during these times. So let's get past all that and let's get over here and start focusing on chapter 26. So let's focus on this chapter dealing with digestion. So first off, let's start talking about digestion and what exactly digestion is, and then we can move forward from there. First off, digestion is the process of taking big macronutrients, things like big carbohydrates and lipids and proteins that we eat, and breaking it down into its simplest components, its subunits, those monosaccharides for carbohydrates, those amino acids for proteins, and those fatty acids, or actually triglycerides, for lipids. So that's what the digestion component does. But then digestion is not simply just a breakdown process, it's also a process of absorbing those nutrients into the body, taking what we break down into those monomers and absorbing it into the bloodstream, into the body, so that we can actually use it for metabolism. And that's how it leads into our next chapter. After this, chapter 26 is chapter 27, which is metabolism, and it just kind of finishes up what we're going to be starting here in this chapter. So digestion and absorption is really kind of the key. Whenever we talk structurally, whenever we talk anatomically about the digestive system, I want you to realize the digestive system really has two main components, and that's going to be the digestive tract or the gastrointestinal tract, GI tract, and also accessory structures. So I'll point that out here in just a second whenever we get to another slide as well. But it's a piece of information that I like for you guys to have to begin the class, because we have a tract that starts with the mouth and it runs all the way through the body. It goes back to the pharynx and down the esophagus. It expands to create the stomach, and then it enters the small intestines and then the large intestines before it exits out of our body at the anus. So that is the tract itself. And plugging into that tract, or kind of attaching to the tract, projecting out of the tract, are going to be the accessory structures. So in places like your oral cavity, we're going to have teeth, we're going to have tongue. Even the roof of your mouth, your palate, is somewhat of an accessory structure because it helps to kind of shred and compact food while we chew. Our salivary glands are going to produce enzymes and mucus and other water that's going to help with this entire process. So is our liver. Gallbladders related with the liver, creating bile that's going to help break down lipids in our digestive tract. And also the pancreas. We're going to find out the pancreas is going to produce all major enzymes that can break down any nutrient that we can eat and utilize in our body. So after we get past some basics of just kind of, functionally what's going on and structurally what's going on in general, let's kind of get into this and let's look at it. So first off, the digestive system, it does provide that means to break down those ingested nutrients. That's what I talked about. The nutrients, I want you to understand, are something that we're going to call those subunits. So those subunits, those monosaccharides, those amino acids, and those triglycerides or fatty acids, those are going to be our nutrients. And this is really what we're going to absorb into the body. Now, once we absorb it into the body, I don't see this term here on this slide, but I want you to know this term. Once we absorb it in, then we're going to convert it into what's called a metabolite. A metabolite, metabolite, metabolite. And so that just means that now this nutrient is inside of our body, this chemical is inside of our body, and it's going to be undergoing chemistry. It's going to be undergoing some sort of either breakdown to create energy, or we're going to use it to rebuild something or build something new in our body. And so that's really where metabolism comes in, and that's going to be our next term. Again, whenever we eat these are unusable in their original form, and so as a result, we're going to need to break them down and then absorb them. Okay, so it's going to, again, the digestive system includes organs that are going to be involved with this. We're going to have the upper and the lower gastrointestinal tract, plus the accessory organs, like I said, are going to be the two main kind of anatomical components. Again, upper versus lower, we haven't talked about this yet, but go ahead and put stomach. At the end of the stomach is where we stop the upper and we begin the lower. And so once we hit the intestines, your intestines are basically your lower GI tract, and above that, the stomach and everything above is going to be your upper GI tract. Now, when we're looking at this, first off, we got 6 main functions of the digestive system. You guys know how this works. Usually we talk about kind of functions, we talk about characteristics, what makes it different than other systems. So here's kind of the 6 main functions. The first function is ingestion. Ingestion is simply putting materials into your mouth, putting solid or liquid nutrients into the oral cavity. And this is the first step of digestion. Ingestion is one of the only voluntary components of the process of digestion. We get to pick and choose what we put in our mouth and if we swallow it, and at that point on, it basically proceeds involuntarily. And so ingestion is really kind of the only voluntary component that we have, voluntary function or voluntary step even of digestion. After ingestion, we're going to start to see motility. We're going to start to see smooth muscle. Don't forget, this is not in the heart. This is not connected to a bone. So these muscles are types of smooth muscles. So the smooth muscles that line the digestive tract, and we're going to talk about that here in just a second. The smooth muscles that line the digestive tract are going to be there to help move food through the tract, but also in places like the stomach where we need to experience churning for different reasons to help break apart the kind of food materials and help with the process of digestion. Then we're going to use those muscles there as well. And then we have secretion. So secretion, obviously I've already mentioned salivary glands and the pancreas and the liver. So other glands that are going to produce secretions, a lot of secretions are mainly water, but in that water, we're going to have different things that are going to help with the breakdown of the nutrients. So we're going to have things like digestive enzymes, and enzymes are going to become very important. We're going to talk about those here in just a second. So these digestive enzymes, these guys are going to help break down those chemical bonds, take those big macromolecules and turn them into those individual subunits, those nutrients that we can absorb. For proteins, we're going to have to use hydrochloric acid. We're going to have to use an acid that's going to help to rip apart those chemical bonds. So proteins are a lot tougher to break apart, and so we're going to have to start using something to do that. As I mentioned before, bile is only used for fats, and basically what bile does is it takes a big drop of fat, because in our body all the fats are heated up, and so they're all melted, and so it's going to take a big drop of fat that's floating through our digestive tract, something that we ate, it heated it up, and now it's kind of just floating as a big drop, and it's going to take that and it's going to break it down into a whole bunch of tiny little droplets. A big drop of fat, we don't have a chance to let those enzymes work on that big drop of fat. But if we use bile, which is called an emulsifier, and that's what an emulsifier does, is it breaks down fat into smaller droplets so that it can be easier to digest chemically. And so we're going to talk about that in a lot of detail. Also, before I leave that, let's don't forget that if there is an opening, a passage to the outside environment, then that's going to be lined with mucosa. And so the number one secretion, it starts at the mouth and it ends at the anus, it goes all the way through the digestive tract, is going to be mucus. We're going to see that's going to be super important, especially in places like the stomach, where if we don't have mucus, then that hydrochloric acid might actually eat through the stomach itself, and that would be a bad thing. And that's actually what ulcers are, is that hydrochloric acid eating at your stomach lining. Now, we have two different forms of digestion, and this is really kind of the main function that we think about for the digestive system. The first is called mechanical digestion. The second is chemical digestion. Mechanical digestion simply just means to rip and shred, to chew it, to rip it up, and then that way we expose more surface area for chemical digestion. It's the chemical digestion that actually breaks the bonds and creates those little subunits that we can absorb. But in order to get to those, sometimes we need to rip and shred, for example, that protein. It's a lot better to rip and shred that steak so that then our enzymes and that acid in our stomach can attack all kinds of different sides of that nutrient instead of just swallowing it as one big whole bite. Okay, so mechanical digestion rips and shreds, it exposes more surface area for chemical digestion, and then chemical digestion is usually undergone by those enzymes. So those enzymes come in and they're going to start to break down those bonds and start to release those individual subunits that we can use for metabolism. The next function, as I mentioned, after digestion, the digestive system, nothing if we didn't absorb those nutrients in. The digestive system is kind of a tube. It starts at the mouth, it ends at the anus. So it's kind of really a tube that's exposed to the outside. And so in order to get food in, we actually have to absorb it through that tube and get it into our bloodstream. So whenever we talk about absorption, that's what we're talking about, getting it into the blood. But we're going to find out that lipids, our bodies kind of evolved to not really digest, not really absorb lipids that efficiently. And we're going to see that. We don't absorb lipids like we do carbs and proteins. Carbs and proteins can go straight into the bloodstream because they're water soluble. But again, fats are not water soluble. So oil and water doesn't mix. So it's harder to get the fats in. So we actually have to sneak the fat in. We have to combine it with a sugar first to make a triglyceride, and then we have to combine it with a protein to make something else. We're going to call it a chylomicron eventually. And so the lipids, we have to sneak into the lymphatic system, which is eventually not a good thing because it allows those lipids to kind of float through our body, and our blood vessels clog those vessels faster because those lipids haven't been adjusted by our liver, and we'll talk about that. Elimination. So the last step is elimination, and this is the process of getting rid of the waste. So again, whenever we talk about elimination, this is the process of compacting the leftover material, and I want you to focus on the word indigestible. Anytime we talk about elimination, focus on the word indigestible. These are things that you put in your mouth, you swallowed, but your body doesn't have the ability to break down into something that's usable. And so because we couldn't break it down, it's going to remain in the digestive tract, and that's what's going to exit the digestive tract as our waste product, as feces. So elimination is really getting rid of that indigestible material. So now that we've gone through some basics, again, here's going to show you the digestive tract and those accessory organs. So the tract starts at the oral cavity, goes down the pharynx, the esophagus, into the stomach. The stomach's just a kind of a ballooning of the tract. Then we hit the duodenum, the duodenum, into the small intestines, into the jejunum and the ileum, and then into eventually the large intestine, cecum, ascending colon, transverse colon, descending, sigmoid colon, out past the rectum, out of the anus, and that is the entire tract. Along the way, we see teeth, we see tongue, we see salivary glands up in the oral cavity. We also see that liver and gallbladder and that pancreas down there in the abdominopelvic cavity. So pay attention. We don't really use the thoracic cavity for digestion. We have kind of just a highway, the esophagus, that's taking food from the mouth, from the oral cavity and the pharynx all the way down to the stomach. But we don't really use the thoracic cavity for digestion. Enzymes. Let's talk about enzymes real quick, and then we'll really start to get into the meat of this thing before too long and chase it through the system and learn what's going on. So enzymes, these guys are going to end in the letters ASE most of the time. So most of your ACEs are going to be types of enzymes. There are some enzymes we're going to talk about. For example, pepsin in the stomach. It doesn't end in ACE, but it is still an enzyme, and it's going to help us break down protein. So these enzymes are going to end in ACE. Enzymes, here we can see that they are catalysts. That means that they speed up reactions, but I also kind of like for you to understand that they make reactions happen when they usually wouldn't. So it's not just that they speed them up, it's that, if many times a reaction wouldn't take place, a breakdown reaction wouldn't happen, a food item won't digest itself while it's sitting on a plate on a table, but whenever you put it into our body, we can add these enzymes, and it's going to make a reaction happen when it usually wouldn't. It's going to break that food item down into those usable units so that we can undergo metabolism. So they are kind of catalysts, and they are kind of making reactions happen when they usually wouldn't. So it's kind of two different definitions, and both are correct. Now, whenever we talk about these enzymes in this class, we're going to have two word named enzymes. So whenever we look at the names of these enzymes, they're going to have two words in their name. And the first word is going to tell you where it is created. So for example, salivary or pancreatic or lingual. And so those words tell you that it's either made by the salivary glands or made by the pancreas or made by the tongue. And then the second word is going to tell you what it breaks down. Again, the second word is what usually ends in the ASE. And then that second word, it's also going to have that prefix is going to let us know what it breaks down. So for carbohydrates, and this is very important, you should go ahead and write this down. Make sure that you don't forget this. Carbohydrates, the big enzyme, the one that takes the big carbohydrates and breaks it down into trisaccharides or smaller. That means either 3 simple sugars linked together, 3 monos linked together, 2s linked together, which are called disaccharides or monosaccharides. That enzyme is called amylase. So the first one that deals with carbohydrates is called ACE, and it ends with 3s or less. Now, the whole goal is to get ones, to get the individual monos, and that's what we can absorb into the bloodstream, into the body. And so this amylase doesn't finish the job necessarily. So we'll talk about that. Well, and I'll go ahead and mention it though, that what finishes carbohydrates are going to be brush border enzymes. These are going to be small intestines. Small intestines look like the border of a brush because they've got all these little fingers sticking up that kind of look like brush parts. And so on the tips of them, there's these finishing enzymes, and that's what I want you to connect with brush border. They're going to finish. They're going to break the 2s and the 3s into 1s so they can be absorbed. And so we'll mention those later on. And brush borders are good for both carbohydrates and proteins. Lipids don't use brush border enzyme, but carbs and proteins do. Now, the next type of enzyme is called a lipase, Lipase. Again, amylase just a second ago was AMYLASE. And that was the one for carbs. Now we're dealing with lipids, and the one that breaks down lipids again is lipase, Lipase. Lipase is basically the only lipid enzyme that we're going to use. We're going to use bile to help break the fats down into smaller fats, and then we can use the lipase to break down those fats into basically triglycerides, because that's what our body can absorb. It breaks it down into fatty acids, but then eventually we're going to link them together to make these trigs, these triglycerides, so we can absorb them into the body. For proteins, we're going to have a few different enzymes. Proteins, the main enzymes that take the largest proteins and kind of break them down into kind of half-size or medium-sized proteins. Those are going to be called proteases, Protease, protease. And then what takes those medium size kind of proteins, the ones that have already been broken down by the proteases, those are going to be called peptidases. So peptidases, Peptidase, peptidase. These are going to help break down the medium-sized proteins into groups of 3 amino acids or less. So kind of similar to what amylase did for carbs. It's going to end with tripeptides, dipeptides, or individual amino acids. And again, the goal is to get individual amino acids so that we can absorb them into the body. So finish the protein digestion, we're going to end up using brush border enzymes, just like we did with the carbs. They're going to be different types of brush border enzymes, but we're going to be using some in the intestines, and they're going to be finishing the job. They're going to cut the tripeptides and dipeptides into the individual amino acids, and that's going to help get that ready for us to absorb. So be ready to see those terms over and over again. So don't forget our different types of enzymes. If you need to hear that one more time, just go back a little bit and rehear that slide, because that's an important slide. Next, let's move on. I know that I don't have this here, and I just have this term muscularis externa on this slide, but I want now for us to talk about the layers of the digestive tract. Unfortunately, I'm sitting here without my textbook, so I don't have the page number and I don't have the figure number. But if you look in your chapter 26, somewhere close to the front, you're eventually going to see an image of the digestive tract by itself, kind of like a little tube. And then they're going to kind of show you a cutaway view of the tube, and they're going to show you the layers of that tube. We also see this in lab. And so that tube, that is our digestive tract, and there are 4 layers that create that digestive tract. The outermost layer, so I'm going to go from outside in. The outermost layer is kind of skinny, and it's really more like a capsule for an organ, and that layer is called the serosa. So again, outer layer, serosa, Serosa. And so the serosa is the outer layer. It's really just a covering. Right inside of that layer is the layer that we see listed here on this slide, the muscularis externa. We don't put it in parentheses usually. This textbook does, but most people don't. The muscularis externa is the layer of smooth muscle. Now, this is what's responsible for moving food through the tract and also for causing what we call segmentation or churning. And I want you to know that there is an inner circular layer of this muscularis externa, kind of circles around the tube and it pinches the tube shut. And then there is a outer longitudinal layer, and this layer kind of acts like an earthworm, and it kind of bunches the digestive tract up a little bit. And whenever we pinch, and then with the circular, and then we kind of push with the longitudinal, it kind of helps to move food through the tract. This is how we're going to propel that food through the tract. And we kind of see that term right here, peristalsis. Let's go ahead and talk about it. So peristalsis is the process of moving food through the tract. A lot of times you'll see the phrase wave-like contractions. So it is kind of a wave-like contraction, moves food through the tract from the mouth all the way to the anus. Now, also in some areas, and definitely perk up a little bit here, in some areas, specifically the stomach and the duodenum, mainly the stomach, has an extra layer of muscle, and that layer is called the oblique layer. And so the oblique layer, which is diagonal, not going around and not running the length, but running diagonal, this actually is what causes the churning, and we call that segmentation or mixing. And so churning, that's going to help to rip and shred. It's also going to help to mix all the contents of this together to make sure that digestion is going to be as complete as possible. Now, the layer above this muscularis externa is called the submucosa, because it is below the mucosa, the innermost layer, the layer that the food is going to touch. The submucosa, the main thing I want you to focus on, has blood vessels, and these blood vessels are going to take the nutrients that are absorbed, and also lymph vessels. So it's going to take all the nutrients that are absorbed, it's going to move them directly to the liver mainly. So most of those nutrients are going to go to the liver, and there the liver is going to act like a bank. It's going to store the nutrients, and it's going to allow them to be released to the bloodstream when we haven't eaten for a while and we need to up our nutrient levels. So whenever we have high levels, the liver stores it, and whenever we have low levels, the liver releases it. So that's really the submucosa with its vessels. The mucosa is the innermost layer. This is actually what the food is going to touch as it's rolling through that digestive tract. So this is the layer where absorption is going to take place. We're also going to see that it's called mucosa because, again, it's going to make mucus from point A to point B. From the beginning to the end of the digestive tract, we're going to have mucus made by that mucosa. So again, here this slide focused on the movements. Definitely don't forget that there's two main movements, peristalsis and segmentation. And again, peristalsis is the wave-like contraction. Segmentation is another term for churning. I don't really care if you worry about this mass movement that's listed at the very bottom. Here's an image that just kind of shows us this process of peristalsis, and then it even shows us the process of mixing and how that oblique muscle layer causes that material to kind of go back on itself and cause that churning. Now, let's talk very quickly about the regulation of the digestive system. Of course, anytime we hit regulation, we're talking nervous and endocrine control. And so, first off, we can talk about the different receptors that are going to trigger some of these nervous reactions. So, some of these are going to be mechanoreceptors. And whenever we talk about mechanoreceptors here, we're talking mainly about some stretch receptors, some baroreceptors. And this is going to allow us to detect the pressure in the system inside your stomach. For example, if the stomach stretches, then it should send a signal to the brain to stop eating because you don't want too much food in your stomach. That can cause some issues if you're overfull. Same thing when waste product, when feces reaches the end of the digestive tract, and it starts to stretch the rectum, then that's going to trigger the defecation reflex, your urge to go use the bathroom to let that out. So stretch receptors are the mechanoreceptors. The main ones are chemoreceptors. These are going to detect the chemical contents of the inside of the tract, which again, the inside of the tract is called the lumen. And so the chemoreceptors are going to kind of make sure things are digesting to the point they need to before it's allowed to move forward. So in the stomach, the protein broken down to the proper quantity before the pyloric sphincter is going to open and allow that material to move forward. Again, when we look at nervous control, all that I want you to focus on is that these are all reflexes. Again, these are reflexive. We talked about that last semester. It's a pre-programmed response, an automatic response to an input. And so anytime we talk about this, again, it is autonomic motor. So any of our reflexes are autonomic, and our most important reflexes are really the autonomic types of stuff. So here, anytime we see a change in that pressure or change in those chemicals, it's just going to be an automatic response. It's not something that the brain has to think about. Again, it's a pre-programmed response to just bring things back into balance. When we look at the endocrine side of regulation, then we're going to have three hormones. And I'll go ahead and give you the lowdown about these three hormones here. Gastrin is going to turn the stomach on. It's going to make the stomach do its thing. It's going to make it secrete. It's going to churn. It's going to make it digest. So gastrin is for the stomach. Secretin and cholecystokinin. Most people call cholecystokinin, which is that last one, they call it CCK, big letters. CCK, that way they don't have to spell that horrendous word there. So secretin, cholecystokinin. I want you to focus with cholecystokinin on the gallbladder, okay? Because cholecyst is a reference to the gallbladder. So this is what's going to make the gallbladder secrete its or release its bile. Secretin is really going to focus on the rest of the small intestines. That's the way I want you to think about this for our class. So secretin is going to focus more on the small intestines, turning on the rest of the secretions in the small intestines and getting the small intestines active, getting it mobile. And we'll talk about these as we move through. Now we're finally at the point where we can kind of see we're starting to enter into the upper tract. So we're going to focus right now on the upper tract, and then on the next presentation, I'll finish this up with the lower tract. So first off, the upper tract begins with your oral cavity, which is the inside of your mouth. We're going to see the teeth, the tongue, the palate. We're going to have those salivary glands, those accessory structures that are going to be involved. This is where mechanical digestion is really the most important. We're going to get most of it there, along with the stomach, the churning. But mechanical digestion with the teeth and the tongue and the palate here are important to, expose more surface area. We're going to get saliva, and one of the things that saliva contains is salivary amylase. So salivary amylase made by the salivary glands, and it's going to break down some of our carbohydrates, remember, down into the 3s or less. And so also in the oral cavity, we're going to get something from the tongue called lingual lipase. So lingual lipase made by the tongue, and it's going to help to break down fats. After that, we're going to enter into the pharynx, which is really just the back of your throat. Not much going on there. It's just when food hits this region, it kind of triggers the swallowing reflex. It gets the conveyor belt. It gets all that peristalsis going. And then that leads us down into the conveyor belt. So the first real part of that digestive tract that's separate from the respiratory, because those other two were kind of shared by respiratory, is the esophagus. So remember, at that point at the laryngopharynx, we have to choose air into the larynx or food and water into the esophagus. And so this is really kind of the first true part. So at the esophagus, whatever we've swallowed, and here's a new term, you can take this down. You see the word bolus, B-O-L-U-S. Bolus is whatever's swallowed. It's just one swallow. Sometimes I say it's a swallowable package, or it's just kind of, one swallow. So after you swallow, that bolus is going to hit down from the pharynx into the esophagus. And again, the esophagus, it's not very crazy. It's just going to make some mucus, and then it's going to transport that bolus all the way down into the stomach. And this is the only component that's in that that thoracic cavity, and so it's really not functioning in the thoracic cavity. Once we hit the stomach, the bolus hits the stomach. We're going to start to mix it with the contents of the stomach. We're going to start to produce the acids and slowly drop the pH, and we're going to start to add some of those enzymes in there. One thing that I haven't mentioned yet is back in the oral cavity, we had salivary amylase and lingual lipase. Well, we chewed the food, we mixed it with those enzymes, and then we swallowed the food and the enzymes. So I want you to understand that the salivary amylase and the lingual lipase, they're going to continue to work into the stomach until the pH in the stomach is going to drop. And I always give the point of somewhere around 4-ish or so. But when the pH in the stomach drops to a certain point, it's going to actually destroy those enzymes. But that's not that bad, because we get about an hour or two of activity from that amylase and that lipase after we swallow it in the stomach until that pH actually drops to the point where it's degraded. And so we continue to break down the carbs and the lipids until we kill those enzymes, and then we turn on our protein enzymes. The protein enzymes work best at lower pH. And so in the stomach, we're going to drop the pH, and then we're going to activate that pepsin, that main protease, and then we're really going to switch and turn off the turn off the carbohydrate and lipid digestion, and turn on and focus mainly on proteins at that point. Once we're in the stomach, we're going to mix that. We're going to drop the pH. We're going to start to degrade it and mix it up. We're going to make something called chyme. We see that term down there, CHYME. So chyme, it is, here's one of my favorite definitions. It's kind of gross. It is a highly acidic, partially digested, soupy mixture of food. So highly acidic, partially digested, soupy mixture, right? And so it's not food anymore. We've got this kind of gross stuff, not really gross, but chyme. It's highly acidic. Unfortunately, if you've ever, and I'm sure that you have, if you've ever vomited, then what you vomited was chyme. You don't really vomit from your duodenum. You don't vomit bile. You vomit chyme, and that's the reason that it can do damage to your esophagus and also to your teeth, to your enamel. So let's look real quick. In the oral cavity, we do have the tongue, and the tongue has taste buds. They talk about papillae, these mounds, and in those mounds is where we have the taste buds. So the tongue is really important for sensory analysis of food. The tongue is going to allow us to detect the 3 T's. I always like to talk about the 3 T's: temperature, texture, and taste, right? If any of those 3 don't check out, then we shouldn't swallow it. And so if it's too hot or too cold, then it can damage the tract, so we don't need to swallow it. If the texture is wrong, if it's kind of mushy, then our body, our mind instantly starts to think that maybe there's some decay involved, and maybe this is not the freshest food. And so we shouldn't put that food in our mouth, because if it's mushy, there's a chance that it could make us sick if it's decaying, if it's not actually edible. And then texture. So excuse me, temp, texture, and taste. Sorry. So then there's taste. Whenever we think about taste, obviously, if it doesn't taste good, then we're not going to swallow it. One of the key things for a lot of toxins and poisons is that they taste bitter. And so we have a natural reflex to kind of avoid those more bitter tasting things. And if we put them in our mouth, a lot of times we tend to spit them out. So the tongue, it's involved with moving food around in your oral cavity. It's also important in the process of swallowing. If you've never noticed, your tongue likes to compress against the roof of your mouth, and it kind of makes a little ramp that forces all the food in your mouth to go down that ramp of your tongue and down into your pharynx, so that we can move it on into the esophagus. So it's not only manipulating and mixing food, but it's also important for swallowing. And obviously it's important for speech. If you've ever heard somebody hold their tongue and try to say their ABCs, they kind of sound like Frankenstein, so go ahead, pause this, do that right now. This is your at-home assignment. So hold your tongue, try to say your ABCs. Are you finished? All right, never mind. So they're very important for speech. And so the tongue, not only for chewing, but also important for speaking. And obviously that's important to maintain society. Secretions, the tongue makes a couple secretions. Of course, when in doubt, everything in the digestive tract is going to make some mucus. So when in doubt, just assume that it's going to make mucus. And then we're going to have that enzyme that I talked about, lingual lipase. So the tongue's going to make that lingual lipase. And as I mentioned, it's going to continue to work after it's swallowed into the stomach until the pH drops. And once that pH drops to a certain point, we're going to destroy that lingual lipase and that salivary amylase, and then we're going to activate that pepsin, that protease. Speaking of salivary amylase, that is in the saliva, coming from the salivary glands. Again, there's 3 salivary glands. We've learned these in lab, the parotid, the submandibular, and the sublingual. You do make 1 to 1.5 liters of saliva a day. Again, this is, think about a 2-liter bottle, so a half of a 2-liter bottle to 3/4 of a 2-liter bottle of spit every day. Sounds pretty gross, but again, you can read most of it is there during mealtime. So whenever we're eating, we need to lubricate that food. We need to lubricate our oral cavity and our pharynx and make sure that we can swallow. And so we do make a lot of saliva. As we can see, most of it's water. So it is really kind of more about this lubrication than it is about simply just a whole bunch of enzymes and different things. We do see this salivary amylase. Amylase is going to, again, break down carbs into groups of 3s or smaller. Again, mucus. And then we see antibacterial. You don't have to know. You can remember the term lysosomes, but remember lysosomes are really just the stomachs of our cells. And so these lysosomes also are antibacterial. They can help to eat and digest bacteria, and so the chemicals that's inside of it. Some of these chemicals end up in our saliva. It's kind of weird, but whenever you cut your finger, many times you put your finger in your mouth, and it's because you naturally know that it's got some antibacterial effects in that saliva. Again, functions of saliva, it's going to moisten that food. It's going to lubricate the food. It's going to help us to also lubricate the oral cavity and make sure that swallowing is going to be efficient. Something that I haven't mentioned is that chemicals in our body, in order for us to detect them at a receptor, then they have to be dissolved into water. So they have to be dissolved into a fluid. So this is the fluid that we dissolve chemicals in so that we can taste them. So if you dry your tongue out and you put food on your tongue, you're not going to taste that food. Because it must be dissolved into water. So this saliva, very important for the process of tasting. It's going to help clean as well as lubricate. And as I mentioned, it's got some antibacterial components. It's got antibodies as well as some of those lysosomal enzymes. Again, saliva, we're going to start to trigger that digestion for carbs and lipids, and we're going to swallow and continue that into the stomach until that pH starts to drop. And so let's don't worry about the regulation too much, but I do want you to know that what makes you salivate. It's not ringing a bell like we learned. We got to link to that, right? We got to build to that. But the key here, the secretions, if you think about food, if you smell food or you see food, then you're going to start to salivate. And so that's one of the things that our society uses to make us go eat food sometimes. You're riding down the road and you smell Burger King. You smell that flame broiled, even though they really kind of microwave most of their burgers. But you smell that flame broiled and you're like, wow, man, that smells good. I bet that burger's going to be good this time. And so it kind of attracts you in and it kind of makes you start to salivate. And once you start to salivate, your stomach starts to churn. And then you're almost at the point where if you don't put some food in it, you're going to not feel very well. You're going to be, that stomach's going to keep churning. You're going to stay hungry and you're going to focus on food. Same thing with commercials, so the sight of food and the thought of food. I always mention Hardee's, how they put these scantily clad people and they kind of link sex also with food. So it's not just that they're showing you this giant picture of a cheeseburger glistening. They're also showing you some scantily clad lady in a bikini trying to get you on two different levels, right? Trying to not only show you that side of food, but trying to work at a different level, almost that sexual level, trying to attract you in based on that. And so we can see, how some of these people are using little dirty advertising to try to stimulate people to come in and just eat their food. And once you start salivating, you almost really want to start adding some food into your body. Once you put food into the body, then you're going to start to salivate. So that's the second one. And the last one is, for example, if you are nauseated. I'm sure you've noticed this, that if you feel nauseated, you feel like you're about to vomit, that your mouth, my daughter she still says this. She says, Papa, my mouth is watering. And so she says that her mouth is watering, and that means that it's time for me to pull over, that she's getting car sick. And so unpleasant, irritating stimulus or something that's nauseating like car sickness, then that's going to start to cause salivation as well. And that's a good way to know, before, if you're going to vomit, you start to have that salivation. Here we can see the parotid gland with the parotid duct. We can see the sublingual gland with its multiple sublingual ducts, and then we can see the submandibular gland with its individual submandibular duct that's leading towards the base of the tongue at the front. Again, mechanical digestion, the process of chewing, it's properly called mastication. So you can see that there. Mastication is the process of chewing. And I definitely want you to pay attention to that second bullet. It says it increases surface area to facilitate exposure to digestive enzymes. In other words, it rips and shreds to expose more surface so that the enzymes can break that food apart and actually get down to those individual nutrients that we need. It says that no nutrients are absorbed in the oral cavity, but that's wrong. There are some nutrients that are absorbed in the oral cavity. We think about diabetics, or we think about people maybe in cardiac arrest. And so underneath the tongue, you can absorb some sugar. You can also absorb nitroglycerin, and these things can help in those situations. So muscles. We're going to use a lot of muscles. Again, it says required coordinated activities of teeth, lips, tongue, cheeks, and jaws. And so we're going to use a lot of muscles. And here's some of those muscles that we're going to use. You're not really too responsible for those. But if you remember, temporalis and masseter were two that we talked about last semester in lab. We didn't talk about the two pterygoids. Again, let's move on past the oral cavity into the pharynx. Now that we're into the back of the throat or your pharynx, remember this is a communal passageway. This is a shared passageway for food and air. So it's shared between the respiratory and the digestive system. Again, the only key thing that I want to focus on here is that when food touches the pharynx, or it touches your uvula also, which is the back of the soft palate, that it starts to trigger the swallowing reflex. It starts to trigger the conveyor belt to move. And what I mean by that is it starts peristalsis. It starts peristalsis in the esophagus, and it starts getting prepared. It says, hey, if there's food that's touching the pharynx, then it's about to go downhill because of gravity, and it's going to then enter into the esophagus. So let's go ahead and let's get the esophagus pumping. So as soon as that food hits, there's no pause. It doesn't just sit there for a second. It automatically just starts to be fed down into the stomach. So pharynx swallowing reflex is about the only thing that we're going to focus on. The esophagus. Again, the esophagus is what connects the pharynx to the stomach. It's the only component of the digestive tract that runs through the thoracic cavity. And so the esophagus really just carries that food and liquid down to the stomach from the oral cavity. Again, don't forget that word bolus. This is simply a swallowed package. Whatever we swallow is considered to be the bolus. And so it doesn't take very long for things to get through the esophagus. Obviously, here we can see that liquid's going to get through faster than solids. Whenever we talk about swallowing, they have kind of changed this up a little bit compared to usual. I've switched it back here, but I'll point it out where I've switched. Swallowing, the proper term is deglutition. As you can see right here, deglutition is the proper term for swallowing. And swallowing has three phases. You can see how I've changed the first phase. They say that the first phase is referred to as the voluntary phase, but traditionally we've called it the buccal phase. If you notice, voluntary is more of a functional kind of way to describe it, and then pharyngeal and esophageal is more of a anatomical, right? So let's stick with one instead of mixing it up. So that's the reason I'm going back to old school. So I like to call it the buccal phase. This is what's when food is between your cheeks. And again, this is the voluntary phase though. So this is where we voluntarily put the food in, and we can make our choice as to what we're gonna actually ingest. So the buccal phase is when the food is in your mouth, in between your cheeks. The pharyngeal phase is when the food is in the pharynx, and this is when we trigger that swallowing reflex. And then it heads on down into the esophagus, and that's in the esophageal phase. And now we're really at that involuntary phase component. So once it hits that esophagus, you're going to have to do some really ugly things to get it to come back. And so at this point, it's involuntary. It's moving on down the track, and it's destined for the stomach. Here we can see the three phases as this lady sings into an apple. She looks really happy. Have you noticed that? She's like, yeah, I love this apple. Sorry. So here's the voluntary phase. And the voluntary phase is also referred to as the buccal phase, because here we can see that the bolus is in between the cheeks, or it's in the oral cavity. And then it enters into the pharyngeal phase. And in the pharyngeal phase, we're going to start to trigger that swallowing reflex that leads us down into the esophageal phase, which ends in the stomach. Speaking of stomach, let's focus on the stomach to finish up this recording and this lecture, and then we will move on and to the lower GI in the next presentation, the next recording. So when we look at the stomach has got some basic components, and we do this in lab. But here's the esophagus. The first component is called the cardia, and then we've got this fundus, and we've got the body, and then we have the pylorus, and that leads into the duodenum. So out down here is the duodenum or duodenum, and that is the first part of the small. So basically the stomach stops here, and it starts somewhere around here. And so when we this, I want you to understand that the beginning and the end of the stomach are really going to focus on mucus to make sure that what's happening on this main pouched region is not going to affect everything else. So whenever we drop the pH in this region, this mucus is going to help to buffer to make sure that the other components, the esophagus and the duodenum, are not going to be burned or eaten by that pH problem, okay, because we've dropped it for our benefit, but it's a problem to other places. The fundus in the body is where we're going to do the churning, and this is where we're going to make the enzymes and the acids that are going to break down those proteins. So the stomach, we're going to saturate the food with gastric juices. I don't like the term juice, but that just means everything that's made by the stomach. So we're talking about the stomach acids, which I definitely want you to know is hydrochloric acid, big H, big C, little L, HCl. And then we're also going to use that main protein called pepsin, and we can see that term there, P-E-P-S-I-N, pepsin. So the stomach, it does both chemical and mechanical digestion. Chemically, it focuses on proteins. Mechanically, it's going to churn. It's going to help to rip and shred, and it's going to help to mix and kind of get all those enzymes and acids bathing all those foods so that we can rip it apart. Food can spend 2 to 6 hours. It's not that crazy important. The facts are, what's more important is the more protein that's in the meal, the longer it stays in the stomach. And that should make sense if this is where we're going to digest the proteins. Then the more proteins we have to digest, the longer that it's going to take for us to digest them. There's little to no absorption in the stomach, but I do want you to know two things that are absorbed in the stomach. Two A's, alcohol and aspirin. So alcohol and aspirin are absorbed in the stomach, and this is a good and a bad thing. When aspirin, whenever we need a dose of aspirin, it doesn't take very long because it can eat through that mucus and get absorbed very quickly into some of the capillaries in the stomach lining. And so that's a good thing to be able to get that medicine in very quickly, but it's a bad thing because if you take too much aspirin, then it eats too much of that mucus and it starts to eat away at your stomach lining. It allows the acids to get down into the actual lining of the stomach, and that's what causes ulcers. So definitely not a good thing. Same thing with alcohol. Alcohol, it erodes that stomach lining. And so if you're a chronic alcoholic and you drink way too much all the time, then you're eventually going to start to have serious ulcers in your stomach due to this, because it eats away that mucus and then allows those enzymes to attack. Definitely not a good thing. The lining of the stomach has these little indentions in it. I'm gonna go ahead and switch over here and show you this. The lining of the stomach has these little pits, these gastric pits, and so you can see these little indentions. Here's the main surface of the stomach where the food's gonna touch. But here we see all these different cell types. So there's all kinds of different cells. These are all the gastric glands that are down in the gastric pit. Here we can even see the layers I was talking about before, the serosa. We see the circular and the longitudinal layer. That's usually all that we would get in most of the lining of the GI tract, of the digestive tract. But here's that extra oblique layer. As I mentioned, that oblique layer is what really causes the churning. Submucosa, we see a bunch of vessels that are going to transport the nutrients away, and then up here is that mucosa. So the mucosa has these gastric pits, and in those gastric pits is where we're going to have the gastric glands, and those gastric glands are what's going to create all the enzymes and those acids. Okay, so there's that slide one more time. There's that image one more time. And now let's talk about what's happening in those gastric glands and in those gastric pits. I don't care if you know the difference between surface and neck mucus cells, but the facts are that there are cells that are going to make mucus, and these are going to be towards the top of the gastric glands, and that's a good thing because it's kind of have to protect most of that lining from what's coming out of those pits, out of those glands. So either on the surface or in the neck, we're going to be there trying to help buffer that pH a little bit and help protect with that mucus. What's really important are going to be the parietal and the chief cells especially. So the parietal cells, I want you to remember this because we don't talk about it hardly ever at all. They produce something called intrinsic factor. So intrinsic factor is very important. What it does is it grabs a hold of vitamin B12 as it's going through your intestines, and then it walks it into your bloodstream. So it helps with absorption of B12. B12 important for your red blood cells to do their normal thing, and so intrinsic factor B12 absorbed into the intestines. What's really important about these parietal cells is they make that hydrochloric acid. They make hydrogen and they make chloride and they mix them together, and then it produces hydrochloric acid. This is what's going to drop the pH in the stomach, and by dropping the pH in the stomach, that allows us to break down those proteins efficiently. Okay, so we've got a couple of functions of this hydrochloric acid though, besides that protein breakdown. We can see starting at the very bottom helps to denature the proteins, and that's what I'm talking about to begin with. Denatures proteins and facilitates chemical digestion. It also helps to break down other components like cell walls in plants, which are very tough for us to break apart. With our normal enzymes, we don't have enzymes that can break that apart, so we need to physically break it apart. Also, the connective tissue. Don't forget your mycelium layers that we talked about. Remember, whenever you're eating a chicken breast, you have the little films in there sometimes that wrap around some of the parts of the chicken. And so those tissues, as we eat them, they wrap that meat up. And if we don't rip through those, then we're not able to actually access the nutrients in that meat. And so it helps to break that down. Something else is it kills most microorganisms that come into the stomach. So if you have a cold and you are moving mucus towards your mouth and then you swallow it, well, then that bacteria or that virus that might be in your lungs is going to enter the stomach, and we're going to start to kill that right there. So it's not going to spread, hopefully. This is good also for whenever you eat things that might have organisms on it, obviously, that can cause havoc in our intestines. And then another super important function is the one that we see up here towards the top. It converts the inactive enzyme pepsinogen. We're about to find out our next cells are going to make the pepsinogen, but they work with these cells and their hydrochloric acid to activate the pepsinogen into the active enzyme, which is actually pepsin. And so pepsin is the enzyme that's going to break down the material and actually break down our proteins and make what we our important protease is what pepsin is, and so it's going to break these proteins, big proteins, down into more medium-sized proteins, so then we can hit them with some of those peptidases. So again, chief cells, they're going to make pepsinogen, and so these chief cells, they're making this pepsinogen, and this is the enzyme, and it must be activated with hydrochloric acid. So basically pepsinogen plus HCl equals pepsin. That's the way you can think about it. Once we get to this point and we turn the pepsin on, we activate the pepsin, now we've activated protein digestion. So this happens when the pH drops to a certain point. At that point, we destroy that salivary amylase, that lingual lipase we talked about from the oral cavity, and then we activate that pepsin and it starts to digest the proteins instead. I don't want to focus on this, but there is something referred to as gastric lipase that's in those chief cells that's being made. We don't really understand its role that well, but it may be involved with fat breakdown. But again, the focus in the stomach, I want you to make sure that you know, is on the proteins. There's another group of cells called G cells, and these G cells, they make the hormone gastrin. And don't forget, gastrin is the hormone that's going to really turn the stomach on, turn on its secretions and its mobility, its ability to kind of do its churning. Here's a little slide, kind of demonstrate. Here's the surface and the neck mucus cells, and then we got all these other cells kind of scattered amongst the rest of that pit. Here's a slide kind of showing you how that hydrochloric acid is working and kind of creating pepsin and all that. You don't have to really focus on that slide though. Real quick to finish this up, talking about the regulation of digestion in the stomach. So stomach, again, is really kind of a pouch. And it's going to create its own kind of movements dealing with this smooth muscle. So it is kind of autorhythmic. It spontaneously depolarizes. So here's what I want you to focus on, though, are going to be the 3 phases of regulation of digestive processes in the stomach. The first is cephalic, which refers to the head. So either food is in the head or you're thinking about food. The gastric phase is when food is in the stomach, and then the intestinal phase is when food has left the stomach and is now into the intestines. So let's move and let's talk about the cephalic phase. Again, the thought or the sensation of food can trigger this. Actually, food entering the mouth can start to trigger this as well. But usually we're talking about just thinking. This is going to trigger some of those higher level regions in the brain, and it's going to start to trigger the stomach eventually to start growling. So again, this is what those commercials and writing by Burger King does, is they put that thought or that sensation of food in there, and now triggered the first stage of the stomach. So now the stomach wants to continue its stages. So once you start to growl with that stomach, it's wanting to move to the next phase. So it wants you to put food in. So it's almost, they're trying to trick you into eating even though you don't need those calories. The second phase is the gastric phase. Food enters into the stomach. There, we're going to start to pay attention with those chemoreceptors. We're going to detect the protein concentration and those pH levels. We're going to release gastrin. As we can see down here, the release of gastrin is initiated by food in the stomach. And so once we release that gastrin, it's going to trigger the statement above it. It's going to increase the mobility and the secretions. Whatever the secretions, the hydrochloric acid and that pepsinogen, it's going to increase those secretions. And so really gastric phase is going to focus on making sure the stomach is as active as possible while the food is inside of it. Once the food is left, then we're going to trigger the intestinal phase. And the intestinal phase is triggered by triggering some of these other hormones, CCK and secretin, cholecystokinin and secretin. They're going to turn off the stomach and they're going to turn on the intestines. So once we leave the stomach with this chyme, with this partially digested soupy mixture, highly acidic soupy mixture of partially digested food, then we're gonna start to trigger this reflex where we turn off the stomach and we turn on the intestines. Okay. At that point, we're gonna move into the lower GI for our next conversation, which will be coming up very soon. I know this has been crazy, but hopefully you are able to follow this with your notes and with study questions and other things, and you are able to get a grasp on this information. You can go back and re-listen to it as many times as you want, and I hope that you have a great rest of your day. And if you have questions, we will be doing a Blackboard Collaborate session during our normal class time, and I will hit some of the tougher topics during that. I can pull up any of the information that I've shown you here today and re-talk about it, or I can just answer any questions that you've got. So otherwise, I hope that this has been fun, and I'll see you soon.